All right, terrific. Thank you very much, and certainly appreciate the opportunity to address this event. So my name is Shlomi, and I am the founder of Eleusis. And today, in the immortal words of Monty Python, now for something completely different, uh, because
Audio is great, by the way, Shlomi. Just confirming.
that's right. So we're going to be talking about the opportunities beyond psychiatry, specifically psychedelics as anti-inflammatory medicines. And we're very excited about this development at Eleusis. And so before diving into it, just giving you just a brief background about the company and what we're doing. So, our focus is to transform psychedelics into medicines, full stop. And we were founded in 2013, so that makes us the first and oldest company in this space. We identified very early on opportunities both within psychiatry and outside of psychiatry. And so Eleusis' mission is to develop the full spectrum of therapeutic potential associated with psychedelics. And psychedelics as a term is a bit of a misnomer, obviously, because in their primary application in anti-inflammatory use, they would be subperceptual. They would not have the psychoactive effects that are so closely associated with serotonin 5-HT2A receptor agonists. Now, Eleusis can also be thought of as the most expensive and overwrought master's thesis of all time. So to give you a sense of where the origin of this came from, I was at NYU. I came across the Johns Hopkins Roland Griffiths paper 2011 about the long-term effects of psilocybin. And I was fascinated. And being the old man at grad school, I could pursue my curiosity. And I just read everything that I could find on the topic. And first, the psychiatric opportunity was obvious, right? And it was already well established in the 1960s. There were meta-studies confirming these results. And it was clear that this was the distressed asset of psychiatry. It was something that everyone thought was worthless, but if developed correctly, could be of tremendous benefit to addressing unmet needs within psychiatry. But there was much more, because outside of psychiatry, the serotonin 2A receptor, and in general, the use of psychedelics has some profound potential. And so the way I would frame the opportunity is that there is the psychiatric, the visible part of the iceberg, which is massive, but there's something beneath the surface, something subperceptual, something that most people are not aware of, but something that could transform the way that age-related chronic inflammation is treated. And that's by using these compounds, these psychedelics, in a not-so-psychedelic manner. And so Eleusis' mission is to pursue this particular opportunity, the whole set, the visible and the opportunity beneath the surface. And so Eleusis Health Solutions is enabling broad access to safe and effective psychoactive drug therapies. That's what everyone else at this conference is talking about. That's what we're doing as well. But given the fact that everyone else was talking about that, we can save that discussion for another day. Eleusis Therapeutics, on the other hand, is doing something that no one else is doing. And I think that when the history of this research field will be written decades from now, I think that people will come to appreciate that there was this massive potential that was really obvious if you just scratched the surface of it, if you just looked at all of the indicators that suggested that this particular receptor, these particular compounds could have profound therapeutic effects in terms of treating inflammation and all the disease processes associated with inflammation. Now, I'm just a spokesmodel, right? I am just a pretty face. Really, the people who are driving this are the scientists. And so these are our favorite scientists, the co-founder of the company, Professor Charles Nichols, Professor Tim Foster, both at Louisiana State University, and our science director, and really kind of at the heart of everything that we're doing, Emmeline Mallette, who's driving our preclinical research and drug discovery programs. So I'm now going to summarize some of their findings and works, and I'll do the best that I can in articulating what I think is the relevance from the perspective of treating inflammation and chronic inflammatory disease. So, unlocking the anti-inflammatory potential of psychedelics. Psychedelics target the most widely expressed serotonin receptor in both the brain and the body, right? When people think about serotonin, there's often this assumption that it's some sort of drug associated with happiness or antidepressive effects. But actually, 95% of all the serotonin in our body is not in our brain. It's in our periphery. It's mostly manufactured in our gut. And the serotonin modulates a wide variety, almost a mind-boggling diversity of processes within the body. And the receptor in particular that mediates the psychedelic effects of psychedelics, the profound perceptual effects, the serotonin 2A receptor, is not just expressed in the brain, which it is quite clearly, but it's expressed on all immune cells that we've studied. It's expressed on all major organ systems, and not just expressed in a small, relative to other serotonin receptors, but highly expressed. And in fact, not just expressed, but it seems the expression of the serotonin 2A receptor appears to track the progression of a variety of diseases. It's almost a biomarker in and of itself in terms of its relative expression from a normal state to a disease state in the human body, and certainly in a wide variety of animal models. Now, interestingly, serotonin itself is primarily pro-inflammatory. So elevated levels of serotonin, just like the serotonin 2A receptor expression, are typically biomarkers of inflammation, are biomarkers of a dysregulated process giving rise to a pretty wide diversity of pathobiology, ranging from inflammatory bowel disease, asthma, cardiovascular disease, fatty liver, going all the way to Alzheimer's. And so it's a very interesting area to study, but it's been an area that's been essentially off limits. And everyone participating here knows very well why that's the case. But interestingly, the serotonin 2A receptor has multiple pathways downstream, meaning that depending on the ligand, the compound that binds to the receptor can signal in a variety of different ways. And that phenomenon is termed functional selectivity. And so when serotonin binds to the serotonin 2A receptor, there's a variety of essential cellular functions that are mediated. When psychedelics bind to the serotonin 2A receptor, you have these profound effects. When psychedelics bound to the 2A receptor, when some psychedelics, not all, bind to the 2A receptor, we also observe anti-inflammatory effects. And that's the profound discovery made by Professor Nichols back in 2007, 2008, that has really been the bedrock foundation of our development within Ellucis Therapeutics to transform psychedelics into anti-inflammatory medicines. So, public enemy number one, inflammaging. The inflammaging is the long-term result of chronic low-grade inflammation. As we age, the immune system becomes dysregulated, either because of excess nutrition, i.e. metabolic dysfunction, i.e. being overweight or having diabetes, or just because of the insults of life, stress, viral reactivation, a whole host of kind of factors, which essentially leads to a process by which you have chronic low-grade inflammation that both drives and is driven by aging. And that's the fascinating thing, is that this is not just about disease, and if that wasn't enough, but it implicitly is about longevity. Because if you can successfully treat the dysregulated processes associated with inflammaging, and whether that's dysregulated metabolic function, inflammation, epigenetic expression, etc., you're not only improving the quality of life, but you're also, in all likelihood, extending life. And so the opportunity, right? So this is the Opportunity Summit. So what is the opportunity beyond psychiatry? It is to treat inflammaging via psychedelics. Now, our research, and when I say our, I mean Professor Nichols, Professor Foster, and the preclinical work directed by Dr. Mallet, has discovered that psychedelics treat inflammation. They enhance cell viability. They provide protection to the cell. They normalize insulin response. they modulate mitochondrial function in a way that preserves the cell's viability and enhances mitochondrial function. They enhance neuroplasticity. They attenuate fibrosis that's dysregulated in many disease processes, such as asthma, for example. And they attenuate or completely prevent viral reactivation and replication. Now, these are not just our findings, right? So we have had other labs or CROs replicate a significant amount of the findings that have been made at the labs at Louisiana State University. And so we're very excited about having multiple models of preclinical effect, right? It all suggests this is not about a particular disease system, a particular organ, but this is a potential broad spectrum opportunity across disease types, and the unifying feature being all mediated by serotonin in one way or another. And so what I'm going to run through now is the discoveries that we've made that have set us down a clinical path that we're very excited about. And so I'm going to try to do it in sequential order. I'll skip the kind of initial in vitro studies that Professor Nichols executed, because we have kind of already touched on that, and jump right into the animals. So the first discovery that was made and kind of built upon over a period of about 5 or 6 years was that very low doses of certain psychedelics, not all, have a profound therapeutic effect in animal models of inflammatory bowel disease, asthma, and cardiovascular disease. And crucially, these effects, these disease-modifying effects, were made possible with doses that are highly unlikely to be perceptible, right? Indicating that there is not only a likely acceptable therapeutic margin, a therapeutic index acceptable for development, but prospectively, as we create new chemical entities that have further functional selectivity, that are further biased towards anti-inflammatory effect and away from psychedelic effect, that we can ultimately move into a wide variety of diseases through conventional formulations like a pill, for example. Now, these anti-inflammatory effects were observed in animals, and specifically where we have biomarkers in the relevant tissue indicating that you see reductions in pro-inflammatory cytokines and pro-inflammatory protein expression, where you see changes in the tissue, you see changes in the capacity of the animal to breathe. And curiously, you also see changes in metabolic function, which suggests that like the general umbrella term of inflammaging, these are paleiotropic effects, meaning that there's some sort of change in genetic expression which is fundamentally altering the cell's expression. And it's not just a particular cell type, it's hitting multiple cell types. It could be both the immune system as well as the target tissue. Now, our second discovery is that psychedelics are neuroprotective, and that's not really just the discovery of Eleusis. In fact, there's multiple labs out there that have already demonstrated that psychedelics can exert a profound neuroprotective effect, and specifically, they can keep cells alive in the presence of toxic conditions. So in this particular slide, we show a very small amount of RDOI, which is kind of our test vehicle, the prototypical serotonin 2 receptor agonist, keeping cells alive in the context of a variety of different toxic conditions. DL-AAA is a toxin that's used extensively in ophthalmology. I think the other one was camptothecin, another toxin used. But we've looked at a wide variety of toxicity, hyperglycemia, a variety of other conditions, and very low doses of the drug, 1 nanomolar concentrations in vitro, keep the cells alive. And that is profound, right? That implies a pretty interesting therapeutic potential across a broad spectrum of unmet needs. So that's fine, it's in vitro. What is it doing in animal models of disease that are potentially more relevant, and specifically that can actually be clinically developed today? And so that's ophthalmology. And this is really based upon the breakthroughs of Professor Foster at LSU. So Tim Foster is just down the hall from Charles at LSU. And one day over lunch, I guess they had a conversation about psychedelics and inflammation, and it just so happened that Tim was looking at herpetic eye disease, so essentially a herpes outbreak in the eye. And Charles said, hey, why don't you try some RDOI? And sure enough, the combination of RDOI, this prototypical serotonin 2 agonist, and an antiviral was superior to not just dexamethasone, right, the kind of conventional corticosteroid, which actually exacerbates viral replication in this particular model, but actually better than just the antiviral alone. And so originally back in 2017, 18, we thought, okay, well, this is very interesting. Maybe we can develop this drug to treat viral mediated inflammation in the eye. But it's a relatively small indication. And so we wanted to go a little bit further and see what the other effects are in ophthalmology, where these drugs potentially stay in the eye. And so why were we focused on ophthalmology? Because when you put a drug in the eye, it tends to stay there. And so you have very low plasma concentrations of the drug, meaning a very low likelihood of psychoactivity. And so we said, okay, well, what else does this drug do? And so we looked at allergic conjunctivitis, which is essentially inflammation at the front of the eye associated with exposure to one allergen or another. And in this particular model, it causes a very profound inflammatory state associated with the degranulation of mast cells. Think of mast cells as depots of a variety of pro-inflammatory cell types that gets released when the antigen, when some sort of allergen is sensed. And so in this model, we induce that, but we pre-treat the animal with the RDOI, with a psychedelic compound. And what we see is that very low concentrations of our compound are efficacious in preventing this very profound inflammatory response. And so that's interesting. But the big discovery was made later, because we then said, well, what are the pharmacokinetics of the drug? i.e., when you administer the drug topically, where does it go in the eye? And what we discovered is profound, we think, we hope. What we discovered is that these drugs administered topically can reach the retina at clinically relevant dose levels. Now, this is a very controversial thing in the field of ophthalmology, right? This is about as risque as you can be in ophthalmology, because some ophthalmologists would say that doesn't matter, it's a dead end because no therapeutic it's a graveyard of therapeutic development. Many people have tried, many people have failed. Others say it is the most important area for development in ophthalmology, because the current standard of care in ophthalmology is to essentially inject the therapies that are treating the retina via intravitreal injection. And it's just as pleasant as it sounds. And to treat diabetic macular edema, to treat macular degeneration, you need one of these injections every month, every 3 months. And so Novartis and Allergan and a bunch of companies, their primary R&D focus is, can we create longer lasting injectable therapies? And that's fine. The problem is that 50% or so of patients fail these therapies, either initially or over time. And these therapies are expensive. And there's no topical solution that actually provides disease modification. And so the fact that our compounds can reach the retina at clinically relevant dose levels, at least in the rabbit, would suggest that we have a breakthrough potential to treat retinal disease. And so we tested that. And so what we went into is an animal model of posterior intermediate uveitis, so inflammation in the middle and the back of the eye. And sure enough, we saw that topical treatment with RDOI was preventing inflammation at the back of the eye. It was attenuating a very profound inflammatory state. Now, it wasn't as effective as a mega dose of corticosteroid, but that's not our objective. We're not competing against drugs that are immunosuppressive, because you can't take an immunosuppressive drug chronically. Instead, we are essentially creating a new therapeutic alternative as an immunomodulatory drug that not just attenuates inflammation, but crucially provides retinal protection. Because what we've observed is that by administering the drug topically, we preserve the retina. You can take a look here at this diagram of the degenerated retina versus the preserved retina. And that's the difference between being able to see and being blind, right? Now, in intermediate uveitis and kind of, no one's injecting tuberculosis into human eyes, thankfully, right? And so there isn't this kind of profound inflammation, generally speaking, in humans. On the other hand, we have all sorts of other problems, right? So if you look at the bulk of disease burden in ophthalmology, it's coming from indications like diabetic retinopathy and macular degeneration. And this ties the whole story together because these conditions are, exemplars of inflammaging, right? They are high oxygen consuming organs. You have a lot of oxidative stress. You have a lot of exposure to hyperglycemic conditions in diabetes. You have a neurodegeneration, which looks a lot like Alzheimer's. You have a lot of the same cell types that are affected in Alzheimer's and associated with aging and diabetes. And these are the leading causes of vision loss in the world. And so we think that our therapies may be effective at modifying the disease course of diabetic retinopathy and macular degeneration. Now, our last discovery in this particular area, I promise you not the last discovery in total, we've got a lot of surprises we can't wait to share, is that we have a new chemical entity, a whole sequence of new chemical entities. And the idea was that how can we make our prototype drug, RDOI, safer and more useful in a clinical context? And so based upon the expertise of Charles and his father, David, we were able to come up with a whole series of proprietary new compounds that improve the safety margin of these drugs, i.e. for the same anti-inflammatory effect, having much less potential psychoactive effects. And so we're very excited to take these new compounds into the clinic to evaluate them in terms of safety and potential efficacy in 2022 and 2023. These are new drugs, and therefore they have a longer lead time before they can get into the clinic, but we believe the therapeutic potential is profound, and we're very excited about it. So I could end my presentation there, but I think that one of the criticisms of me, which is absolutely fair, is that I'm very ambitious, right? And so ophthalmology is not enough. There's so much more to do. And so one of the things that we are also looking at that we're very excited about, that we're actually known for, is LSD. Why LSD? Right? LSD is not the most, when we evaluated a whole series of psychedelics, it's not that LSD is the most anti-inflammatory, far from it. There are other compounds that have a far more potent anti-inflammatory effect. However, LSD may be the best drug to modify the cruel progression of Alzheimer's disease. So right now at Johns Hopkins, there is a study to look at psilocybin in the treatment of the neuropsychiatric symptoms of Alzheimer's. Essentially, in the early stages of Alzheimer's, one of the most reliable biomarkers of the progression of Alzheimer's is apathy, depression, and anxiety. And so there's a study underway led by Albert Garcia Romeo and Paul Rosenberg, who's one of the key thought leaders in the field of neuropsychiatric symptoms of Alzheimer's, looking at, does a high dose of psilocybin provide relief from depression and anxiety? And that's great. But that's not exactly a disease-modifying therapy. That's a therapy that may address the symptoms and may actually even show some disease modification. But the question is, what's a scalable approach to providing disease modification in Alzheimer's disease, i.e. slowing or halting its cruel progression? And that's where LSD comes in. So Alzheimer's is, just like retinal disease, it's a great exemplar of inflammaging. And in fact, if you have one of the retinal diseases that we talked about, diabetic retinopathy or macular degeneration, you're at much greater risk for Alzheimer's. So there's a clear linkage, same cell types, a lot of the same underlying causes for the degeneration and the inflammation associated with both diseases. Now, AD is a multifactorial disease. It is resistant to precision medicine. And despite the best efforts by Biogen and a variety of other companies that have targeted amyloid or tau, you can sponge all the amyloid out of the brain, and you can sponge all the tau out of the brain, and you don't really significantly halt the progression of Alzheimer's. And so our perspective is that you need a multifactorial approach. You need to be hitting multiple disease targets in Alzheimer's simultaneously to have any shot at modifying its progression. And LSD does that. LSD has a very unique pharmacology. It hits multiple receptors, including the serotonin 2A receptor. And that combined capacity means that that LSD kind of, it punches above its weight, right? You can administer 15 micrograms of LSD and you can observe profound effects in fMRI, which would suggest that very low doses of the drug are having key changes in the brain associated potentially with a therapeutic process. Now, we've conducted a phase 1 safety study with LSD. Nilafar family, our leader of psychiatric research, oversaw this study. Robin Card Harris was involved in this, and we'd love to talk about this particular study. But essentially, it was just the beginning. Can we show that low doses of LSD are safe in a healthy elderly population? But we extended past that. We believe that LSD hits multiple disease targets associated with Alzheimer's: amyloid, cognition, neuroplasticity, preventing viral replication, metabolic function, oxidative stress, epigenetic expression, you name it. And we are about to release a white paper which articulates the scientific basis for taking LSD into phase 2 trials for Alzheimer's. A recent study at University of Chicago demonstrated that 13 micrograms of LSD has discernible and significant effects as measured by fMRI. So there is clear target engagement, which in drug development is almost like a third or half the battle when coming to treating any type of disease in the brain. And so we're very excited about our pipeline. We are going to be advancing ELE2 into ophthalmology. We are going to be advancing LSD into Alzheimer's, and we are developing new compounds to treat a variety of peripheral diseases like asthma, cardiovascular disease, fatty liver, et cetera, with new candidates that we are developing. So the bottom line, the part of the iceberg that's above the surface, that's great, right? There's a huge unmet need in psychiatry. That's why I got involved in this space, and really kind of a lot of the original reason for starting Eleusis. But that's only part of the story. It's a part of the story that we're going after in a very exciting way, which I'll be happy to talk about at some other time. But we believe that there's a whole other opportunity in inflammation. Massive unmet needs, which are much lower challenge in terms of delivery, because you don't need a therapist. You don't need a special treatment room. You don't need an over elaborate REMS. This can be in an eyedropper. This can be a topical solution. This can be a pill. And so it scales very quickly. And the revenue potential here is massive. And so we're very excited about that, because what that means is that we can raise a lot of capital to move forward the science and to demonstrate the therapeutic potential here in humans. And that's really why we got into this, is to make a difference. And so with that, I'd be happy to address any questions. Thank you.
Yeah, Shlomi, that was brilliant. Thank you so much. A couple of questions here. The first one is, with regards to the drug delivery, so you mentioned the eyedropper as one example. Are you going to be manufacturing the actual delivery mechanism so you can monitor dosing? Are you working with other companies? There's not a lot of, there are some, and we actually have Mike Arnold from Silo coming later on who will talk about delivery for psilocybin, but there's not a ton in the delivery mechanism right now, at least that you read about. So are you doing that in-house? Talk to us a little bit about that.
Yeah, so at an early stage, phase 1, the manufacture of the investigational product, the IMP, is going to be relatively basic. It's just going to be a simple eye drop solution. The question will really be, at what dose do you see psychoactive effects? If it turns out that if you give, let's say, more than 2 times the recommended dose, that's going to require some pretty high-tech way to prevent people from overdosing with that. But if the dose required for psychoactivity, it turns out to be 10 times or 20 times or 50 times the dose that's effective to treat the disease, well, there, I think we're going to be in a conventional space. It'll just be like any other eye drop topical drug, because unless you drink multiple bottles of the eye drop solution, you should not have any type of psychoactive effects. And so we can look to the examples of lorcaserin, Belvic, which was a weight loss drug, to understand what the FDA and DEA guidelines could possibly be associated with the REMS and DEA classification of this type of therapeutic.
Now, thank you for that. Now, I've seen some stories before, but it would be great if you could elaborate a little bit on the relationship between depression and inflammation, particularly, well, maybe it's chicken and the egg, which one causes the other, but what's your understanding of depression and inflammation, their relationship, and then maybe some windows into how psychedelics, one of the reasons why it might be successful in treating the other?
Yeah, well, I think it's a fascinating topic. I think that there are some people like Andrew Miller, Charles Raison at USONA that really spent, years studying the linkage between depression and inflammation. Carmine Pariente at King's College is another kind of thought leader in that particular space. And some of Professor Pariente's research has revealed that there are biomarkers of certain pro-inflammatory cytokines which predict which patients will respond to SSRI therapy, which is very interesting.
Hmm.
So there is a relationship. However, to say that inflammation is the cause of depression or that depression is the cause of inflammation is to radically oversimplify what is an incredibly complex interplay between the immune system and kind of our thoughts and feelings and emotions, et cetera. But the fact that we have, going back to Freud, somatization, right? The fact that you can repress a bad feeling and then it shows up as a rash, right, tells you that there's a clear basis to assume that there's some interplay. And we think that serotonin is really at the heart of that. The same mechanism, the same serotonin modulation of mood may also be implicated in a lot of the disease processes associated with inflammation. You can induce the animal model of depression by injecting an animal with a pro-inflammatory cytokine. It's called animal sickness behavior. And then if you block that cytokine, then the animal's not depressed. And so you can say, okay, well, maybe there's a relationship. So longer term, I think that there will be a lot of biomarkers that tell us which patients are likely to be more responsive to particular types of therapies based upon things like elevated levels of pro-inflammatory biomarkers. But I do think that fundamentally, things like neuroplasticity may factor in much more highly into treating depression longer term than symptomatic treatment. And that's the key. To treat depression, it's not just about alleviating symptoms, it's about facilitating adaptation. And that's what I think Matt Johnson was talking about with Rescola Wagner and behavior therapy. And that's what I wrote my master's thesis on, is this idea that it's not just about symptom relief, it's how do you facilitate learning, right? Good psychotherapy is about learning. And that's another topic for another day.